Hello, everybody. Miss Peachy again from WCA Biology. We are looking at Unit 5, Lesson 12, how cells are used in the body. And this one's going to be kind of short because, again, a lot of repeat and a lot of review going on in this lesson. So as we kind of go through the lesson, they talk a little bit again about mitosis and how mitosis is the process by which a parent cell will copy itself and make exact replicas or duplicates of itself. It's a type of cell division, and it is what's considered to be a type of asexual cell division, meaning that you are not combining two different genomes together. You're just making a copy of one type of cell. And then the kind of cell reproduction that mitosis goes through, we call this proliferation, when cells make copies of themselves, or actually when anything is kind of making more of itself, we say it's proliferating. So it results in an increase in a cellular population. And then to make different kinds of cells, to be able to take like one generic cell and have different kinds of cells, that's called differentiation. And differentiation is how cells become specialized to fulfill different functions within the body. That the process of cell differentiation is one of the things that happens with gene expression. So gene expression is done by decoding some of the DNA that you have. And, there's many different byproducts of gene expression. There's different proteins that are made and different cells are differentiated in this way. And so that's through that process of transcription, translation, and folding of proteins. And then that stem cells are kind of those generic versions of the cells that in embryos, we have two different kinds of stem cells. We have the totipotent and pluripotent stem cells, but in anything other than embryos, we just have one kind of stem cell called multipotent stem cells, and they're capable of dividing and making copies of themselves up to a certain point. And so how do cells know how to make copies of themselves? And this is something that is communicated to the cell through a bunch of different kind of mechanisms. So as the reading kind of specifies, there are environmental influences. Certain things in the environment may trigger cell replication. Nearby cells can communicate information to stem cells to tell them to divide. Chemical signals can affect the cell specialization. So different chemical signals will kind of determine what type of cell that stem cell differentiates into. So sometimes those chemical signals are things that are produced during protein synthesis, during that process of gene expression. Perhaps a hormone or something is made during protein synthesis, and that hormone then influences a stem cell and tells that stem cell what type of cell to differentiate into. But this is known as the cell fate. So the cell fate is determined based on clues in its environment, nearby cells, different types of chemical signals that influence the cell and kind of tell it how to differentiate. Going on in the next slide here, it talks a little bit about cell division again. It talks about how when a cell is no longer able to divide, it becomes a senescent cell, meaning it can't divide anymore. And that time period kind of differs depending on the type of cell we're talking about. For example, in the text here, it says skin cells live for 2 to 3 weeks, and so they are constantly dividing and making new copies of themselves. But colon cells last only 4 days, but some bone cells can live up to 50 years. So the lifespan of cells kind of depends a lot upon the environment and a lot about how, I guess, the type of conditions that cell is exposed to. Certain cells are a little bit have more harsher environmental conditions, and so they need to be replenished and replaced more frequently than others. Your skin is obviously exposed to the outside environment. Your colon is exposed to everything that you eat, and so those cells have a shorter lifespan and need to replenish themselves more frequently. Some cells never replenish themselves. For example, in humans, brain tissue only replicates under very specific circumstances. Some cells in the liver and pancreas can be replaced while others cannot. So the number of times a cell can divide before it's no longer able to propagate is called the Hayflick limit. So this is a different term, and this is one of the reasons why I decided to even have this little video today, is because we have some different terminology in here. So the Hayflick limit is the number of times a cell can divide before it's no longer able to propagate or replicate. We call that the Hayflick limit, and it is named after a scientist named Leonard Hayflick, who cultured different types of cells in his lab. He went and found that cells go through different phases where they're really rapidly replicating and rapidly doing mitosis, and when then mitosis is slowing down, and then a phase where they become senescent, where they no longer are able to replicate and eventually they die. So cells can also die due to injury or poor health of the body, so they don't have to necessarily go through all those phases. If they are injured, they might die without progressing through those phases. And it says most cells are replaced by new cells when they die, but some cells are not. Some cells in your body do not get replaced naturally when they die. So it is untrue, because this whole lesson began by saying, are you replacing your body every 7 years? It's untrue that you are. You are not entirely replacing your body every 7 years. Most of the cells may be replaced, but some cells live longer than 7 years, like bone cells we mentioned, and some never get to be replaced at all. So you don't completely regenerate a new organism every 7 years. It's, I think, fairly obvious when you see people progressing and aging that you can't completely replace yourself every 7 years. So that is the essence of this particular lesson. We'll see you again in the next one.